Overview of mitochondria respiration and stage 2 of carbohydrate metabolism, pyruvate oxidation. There are 4 stages of carbohydrate metabolism. Stage 1 is glycolysis, including glycogenolysis. Stage 2 is a conversion of pyruvate to acetyl coenzyme A. Stage 3 is TCA cycle, tricarboxyl acid, Krebs, or citric acid cycle. Stage 4 is electron transport chain, ETC, oxidative phosphorylation. Mitochondria are specialized organelles responsible for a high capacity for ATP regeneration through the consumption of oxygen via a process called oxidative phosphorylation. This two-term name combines the oxidation of carbohydrate, fat, and amino acid fuels leading to the transfer of electrons in successive steps of oxidation and reduction with the phosphorylation of ADP to ATP. The structure of a typical mitochondrion is presented here. Mitochondria are unique organelles in that they have a double membrane and have a separate nucleus and pool of DNA. The space between the two membranes, the intermembranous space, is where the proton gradient is generated in oxidative phosphorylation. The inner region, surrounded by the inner membrane, called the mitochondrial matrix, is where the reaction of mitochondrial respiration occurs, with a subset of these reactions structurally connected to the inner membrane. For example, the reaction and structural component of the electron transport chain, ETC, occur along and within the inner membrane, and some reaction of the tricarboxyl acid or TCA cycle are also structurally linked to the inner membrane. Notice that the inner membrane is highly complicated in that it repeatedly folds into the matrix region. Such fold, called crista, plural cristae, increase the surface area of the inner membrane, thereby increasing the possible number of molecular transporter and component of oxidative phosphorylation. Interestingly, in tissue known to have a high demand for the consumption of oxygen, such as heart muscle and slow-twitch skeletal muscle, mitochondria have a more intricate network of cristae to support their greater demand for high rate of substrate flux through complete oxidation, which of course require oxygen consumption and along the way produce carbon dioxide. The double membrane of the mitochondria still allows selective molecular transport into and from the mitochondria. The outer membrane is freely permeable to many small molecules and ion, and a transport system allows the inward movement of proton but prevent proton movement from the intermembrane space to the cytosol. Certain molecules can be transported in and out through the inner membrane, such as TCA cycle intermediate, acetyl coenzyme, citrate, oxaloacetate, alpha-ketoglutarate, and malate. Many amino acids can also be transported through the inner membrane, such as glutamine, aspartate, arginine, histidine, and isoleucine, etc. And fat enter the mitochondria via process dependent on their chain length. Short-chain fatty acid, less 15 carbon, enter the mitochondria by passive diffusion. Long-chain fatty acids, such as palmitate, require special transport system known as carnitine shuttle. Thus, mitochondrial respiration is a site within cellular biochemistry that occurs for the complete oxidation of carbohydrate, fat, and the carbon side chain of amino acid. Pyruvate enters the mitochondria in the form of pyruvate through the pyruvate dehydrogenase enzyme complex during low to moderate intense exercise. In the presence of adequate oxygen and where the rate of pyruvate production from glycolysis does not exceed the capacity of the muscle fibers mitochondria to receive pyruvate, the pyruvate enters the mitochondria where it is then oxidized by a series of three enzyme catalyzed reactions. The three enzymes are interconnected in a multiple enzyme complex known as the pyruvate dehydrogenase complex. The overall reaction over here represents the sum of three reactions, as these three enzymes are grouped together into a definite complex, where the presence together is required to support the conversion of pyruvate to acetyl coenzyme A. The multiple reactions are simply summarized into the one overall reaction here and often referred to simply as the pyruvate dehydrogenase reaction. This is how we refer to the complex reaction in this lecture. A new molecule of interest and importance to mitochondrial respiration is coenzyme A. When coenzyme A is combined with acetate group, the molecule acetyl coenzyme A is formed. The product of pyruvate dehydrogenase reaction are carbon dioxide and 2 Na plus proton and 2 acetyl coenzyme A. The CO2 produced means that the 3 carbons of pyruvate have now been decreased to the 2 carbon acetate group, which combine the coenzyme A to form acetyl coenzyme A. The carbon to sulfur bond CS of acetyl coenzyme A has a high acetate transfer potential and therefore readily release the 2 carbon acetate group in the first reaction of the TCA cycle catalyzed by citrate synthase. And glossary words.